Do you want to drink some milk with me?
Sure, but I don't know if I can. Maybe I can try it first.
Why? You can't drink milk.
I don't know, but when I was little, I'm always had a stomachache after I drink milk.
Oh, that's so bad.
Yeah, but strangely, as I grew up, now I will not have a stomachache when I drink milk.
As we just saw in the vlog, my roommate used to have stomach cramps and diarrhea after drinking milk when she was little, but interestingly, she rarely feels uncomfortable with milk now. This common daily phenomenon is actually closely related to a very well-known digestive issue, lactose intolerance. Today, I'm going to explain the biological reason behind it and the key role played by one vital enzyme. To understand why milk sometimes causes stomach aches, we first need to look at how digestion works at the molecular level. Take a look at this diagram. It shows how enzymes work. The molecule being digested, in this case lactose from milk, is called the substrate. The molecule that does the digesting is the enzyme, and for lactose, that enzyme is called lactase. You can see the enzyme has a special spot called the active site. The substrate fits perfectly into it, like a key into a lock, forming what we call the enzyme-substrate complex. The enzyme then breaks the substrate into smaller, usable pieces for lactose. That's glucose and galactose. Crucially, the enzyme itself is not used up in the reaction. It goes back to normal, ready to help digest the next lactose molecule. So how exactly does lactase make digestion happen? The answer lies in activation energy, which you can see on this graph. Without the enzyme, the reaction to break down lactose has a very high activation energy, like climbing a huge mountain. It's slow and hard to get started, but with the lactase enzyme, the activation energy is lowered, like building a tunnel through the mountain. The enzyme creates a different, easier path for the reaction. That means lactose gets broken down quickly and efficiently at our normal body temperature. This graph also shows something very important for our story. When there's a lot of substrate, the reaction speeds up, but once all the enzymes are busy, we reach enzyme saturation. Adding more substrate won't make it faster. This means if you don't have enough lactase enzyme, no matter how little milk you drink, the reaction will be too slow, and lactose won't get digested. Now, why don't we all have enough lactase? This graph shows us why. Every enzyme has an optimal temperature and pH where it works best. For human lactase, that's 37 C, our body temperature, and the slightly alkaline pH of our small intestine. If these conditions change, like if your gut pH is off, the enzyme's shape changes, and it doesn't work as well. But the main reason for lactose intolerance is called lactase non-persistence. For most people, after childhood, the gene that makes lactase turns off. The amount and activity of the enzyme in the gut drops. Without enough lactase enzyme, undigested lactose passes straight into the large intestine. Bacteria there ferment it, producing gas and acids, which cause bloating, cramps, and diarrhea, the classic symptoms of lactose intolerance. So, going back to my roommate's story, her stomach aches as a kid were likely because her lactase enzyme wasn't working at full capacity. As her digestive system matured and stabilized, her enzyme activity became more consistent, which is why she can now drink milk without problems. To better understand how lactase works, we conducted Lab 4, measuring lactase enzymatic activity, which lets us quantify how fast this enzyme functions. We know enzymes speed up reactions by lowering activation energy, and lactase specifically breaks down lactose. But we cannot see or measure lactose directly with a spectrophotometer, so we use OMP, a molecule that looks like lactose and can be broken down by lactase. When lactase acts on OMP, it produces a yellow product, which allows us to measure absorbance and calculate enzyme activity. Our test temperature was 37 C, the optimal temperature for human lactase. Our goal was to measure how fast lactase works over time. We set up reactions and stopped them at 30 seconds, 60 seconds, 90 seconds, and 120 seconds. We also included two controls to make sure our results were valid. Control A had no enzyme, and Control B had no substrate. Only when both enzyme and substrate are present can the reaction happen. First, we extracted lactase from commercial tablets. We crushed the tablet, added PBS buffer, vortexed, and centrifuged to get a clear enzyme extract. Then we added buffer, OMP substrate, and lactase enzyme into each tube. All tubes were incubated at 37 C. At each time point, we added sodium carbonate to stop the reaction, and finally measured absorbance at 450 nanometers. Our results were clear. Absorbance increased over time, and the yellow color became darker. This means more product was formed as time passed, showing lactase was continuously working. The two controls showed no color change at all, which confirmed that the reaction depends on both lactase enzyme and its substrate. The slope of our absorbance time graph represents the rate of enzyme activity. Perfect. Now let's go back to my roommate's story that we saw in the vlog. Why can she drink milk comfortably now, even though she couldn't when she was little? There are two most reasonable explanations, both closely related to lactase enzyme. The first possibility is that her body now produces more lactase enzyme with higher activity. As her digestive system matured and stabilized, the expression and activity of lactase increased. With enough functional enzyme, she can fully break down lactose without discomfort. The second possibility is that the milk she drinks now is pre-treated with lactase. Many dairy products add extra lactase during production, which breaks down lactose in advance. So when she drinks it, no extra enzyme is needed from her body and no symptoms appear. In the end, everything comes back to one key molecule, the lactase enzyme. And these two possibilities lead us to a wider topic. Enzymes are not just important in our bodies. They also have many amazing real-life applications. Just like the lactose-free milk with extra lactase, enzymes are widely used in daily life to make products safer, more convenient, and more efficient. For example, in food industry, enzymes are used to make cheese, yogurt, bread, and fruit juice. In cleaning products, many detergents use enzymes to break down stains like oil, blood, or food residues. In medicine, enzymes help with digestion, wound healing, and even disease treatment. All these uses show how powerful and practical enzyme technology is in our modern life. Finally, I would like to express my sincere thanks to Professor Lee for his guidance and support. I also want to thank my TPS group members, Zumeng An, Ziqian Song, and Liang Tang. Thank you all.